Harvesting the sun, how photosynthesis can turn light, air and water into carbohydrate. Hello, I'm Professor Alison Baker. I'm a plant scientist, researcher and teacher at the Centre for Plant Sciences, University of Leeds. In this podcast, I'm going to tell you about photosynthesis and how plants are the most amazing solar-powered biofactories. Plants use the energy from sunlight to extract electrons from water, releasing oxygen in the process. These electrons are passed on via a series of intermediate compounds, eventually to reduce carbon dioxide to carbohydrate, which can then be converted by the plant into all manner of biological molecules. Photosynthetic organisms are photoautotrophs and are at the base of pretty much every food chain. Before oxygenic photosynthesis evolved, no oxygen-breathing organisms could exist on the planet. Plants don't just produce food. That carbohydrate produced by photosynthesis can be broken down and reformed by the plant into literally thousands of different types of compounds which find use as fuel, for example wood, fossil fuels like coal and oil, fibres such as cotton, linen, flax, construction materials and medicines. Some examples of the latter are anti-cancer drugs like Taxol and vincristine. The process of photosynthesis takes place in organelles called chloroplasts. Chloroplasts are complex and beautiful structures, as seen in this electron micrograph. They are surrounded by a double membrane, termed the envelopes, and have an internal membrane network, termed the thylakoids. The internal soluble space is called the stroma. Photosynthesis can be broken down into two distinct but linked stages. The light-dependent reactions occur in the thylakoid membranes and convert the sun's energy into chemical energy, ATP, and reducing power, NADPH, and releases oxygen. The light-independent reactions occur in the stroma, fix CO2, and use the ATP and NADPH produced by the light reactions to do so. We will first consider the light-dependent reactions. Light harvesting is carried out by molecular machines in the thylakoid membranes. Antenna complexes, proteins with pigments bound to them, trap the light energy and pass it on to photosystems, which use the light energy to remove an electron from a special chlorophyll molecule within the photosystem and replace the electron with one from water. Electrons are used to reduce NADP to NADPH, which is a source of reducing power used in the light-independent reactions. The energy associated with the electrons is converted into chemical energy in the form of ATP. The processes underlying the mechanism of photosynthesis are therefore coupled oxidation and reduction or redox reactions. Remember that oxidation is loss of electrons with or without loss of protons, and reduction is gain of electrons with or without protons. The first step in harvesting the sun is the absorption of energy from photons by pigments. The principal pigments in light-grown plants are chlorophylls A and B, xanthophylls and beta-carotene. Pigments are readily visualized due to their color. Chlorophyll is green and carotenes, responsible for the yellow-orange colors in carrots, are yellow. Pigments can be separated by thin layer chromatography, where a small dot of the pigment or a mixture of pigments is applied at the base of a sheet of plastic coated with silica and a solvent allowed to rise up through the silica by capillary action. The pigments are separated on the basis of their physicochemical properties. In the experiment shown here, pure pigments were spotted as standards and co-chromatographed alongside extracts from dark and light-grown maize plants. Comparison of the pattern shows the presence of all the four pigments in the light-grown maize plants. This slide shows the absorption spectrum of chlorophyll A and B. As can be seen, both chlorophylls absorb strongly in the blue and red parts of the spectrum, but not in the yellow-green part, hence the green colour of chlorophyll, as green light is reflected. When a chlorophyll molecule absorbs a photon of light, the energy can be transferred rapidly and efficiently to nearby chlorophyll molecules. Eventually, the energy is passed on to a chlorophyll molecule associated with a photosystem, which is a complex of proteins and pigments in the thylakoid membrane. The energy of the photons ends up at the photosystem as it is at a lower energy state than the other antenna chlorophylls. The photon's energy is used to oxidize the photosystem chlorophyll. An electron is ejected, leading to photochemical charge separation. This slide shows a schematic diagram of the light reactions with the coloured shapes representing assemblies of proteins and electron transfer components associated with the thylakoid membrane. 4 photons of light are absorbed by photosystem 2. The energy from these photons excites a special chlorophyll molecule within the photosystem. The first photon results in the removal of an electron from the special chlorophyll, that's oxidation, and this electron is replaced by an electron taken from a molecule of water. This reduces the chlorophyll and oxidizes the water. This happens 4 times, once for each photon. The electron taken from a chlorophyll molecule has a lot of energy and is transferred from one component within the photosystem to another in a series of oxidation and reduction reactions, and finally onto the cytochrome complex. As the electron passes through the cytochrome complex, it gives up some of its energy, which is used to pump protons from the stroma to the thylakoid lumen. This builds up a gradient such that there are more protons in the lumen than in the stroma. This gradient can be used to provide energy for the synthesis of ATP using the same mechanism as in mitochondria. This is an ancient process that evolved long before the last common ancestor of animals and plants. When the electrons exit the cytochrome complex, they have lost a lot of their energy, so they need another boost. They travel to photosystem 1, where another 4 photons of light are absorbed, and one after another, the electrons travel through photosystem 1 to reduce NADP to NADPH. NADPH is the source of reducing power, which along with ATP is used in the light-independent reactions. Thus, the products of the light reactions are oxygen, NADPH, and a proton gradient that can be used to drive ATP synthesis. This slide shows the same process as the previous schematic, but displayed in a different way. Here, all the components are shown according to their energy level, more properly their reduction potential, which is a measure of how easily they can give up electrons. This representation is known as a Z scheme or Z diagram because if you turn it sideways, it looks like a Z. Showing the process this way emphasises how light energy is used to raise the reduction potential of electrons within photosystem 1 and photosystem 2. PS2 star and PS1 star indicate the excited forms of the photosystems after light absorption. And so electron transfer proceeds in an energetically favourable or downhill direction, allowing NADPH formation and proton pumping. This is a similar energy diagram to the previous one and shows how chloroplasts can use just photosystem 1 and the cytochrome complex to cycle electrons. This allows a proton gradient to be formed without the splitting of water or the production of NADPH, and is used when chloroplasts need ATP but not NADPH. This process is called cyclic electron transfer. Here's a summary of the light-dependent reactions. Pigments harvest the energy of sunlight. Light energy causes a charge separation in a special chlorophyll molecule in photosystem 2, and the electron is passed on to the cytochrome BF complex. An electron from water replaces the one lost from photosystem 2. 4 photons of light release 4 electrons and result in the evolution of 1 molecule of oxygen. The energy of the electrons passing through the cytochrome BF complex is used to move protons from the chloroplast stroma to the thylakoid lumen. This proton gradient can be used to make ATP, as you'll see in the respiration podcast. Electrons from the cytochrome BF complex reach photosystem 1. Another photon induces photochemical charge separation in photosystem 1, and the electrons go on to reduce NADP to NADPH. When NADPH is plentiful, cyclic electron transfer using photosystem 2 only can be used for ATP synthesis.